I'm just gonna now jump in with the final section. Well, let me check. Other than closing remarks, our last sort of lecture is on the kind of current and future direction of miniature microscopes. We'll end up focusing on what our Miniscope project generally is working on now and in the near future, but also try to highlight what other groups are doing as well. And the morning talks were a great example of this. And really, one great thing about an open source community, and this community in particular, is people are incredibly willing to help each other out. So like for the Miniscope, when Tico's group was starting to develop the Miniscope, we were also kind of towards the end of getting the first kind of prototype of the version 4 working. And so we were able to help each other out, figuring out how to kind of reverse engineer some of the properties of this Python 480 sensor that we both use. And so it's really nice to have this community. And on the analysis side, I think it's true, completely true as well. So it's really nice to have a community that supports each other. But where do we go from here? I think over the last few years, it's become more and more evident that moving when possible to as naturally behaving animals as you can is really important. And there's papers that have come out over these last few years that show just how important it is. So like for a really simple example, years ago, when you would do these types of experiments or do like eye tracking experiments in rodents, you would pretty much always have to head fix them. And then you look at their pupil movement. And the assumption was like, oh, like rodents, because they have such a large field of view, they really don't move their eyes much. And then Jason Kerr's group made this head-mounted, I think it was attached to one of their head-mounted microscopes, but came along with eye tracking image sensors for freely behaving rats. And all of a sudden, you see that like, this is not the case. It's this artifact of head fixing animals. And the moment you let them free, their eyes are going, crazy. And this is true for, properties of just neural dynamics and the types of behavior it can do. So moving towards more and more naturally behaving animals seems to be a trend in the field. And then being able to pull out more and more reliable data that you can look at longitudinally is also extremely powerful. And miniature microscopes are a great fit for that. So before getting into some of our like current work, I just stole this slide from Tico's presentation earlier today, just to highlight like the breadth of development within this one specific area in like tool development in neuroscience, right? So there are definitely commercial versions that have their own value. There's also the other kind of main commercial miniature microscope type device would be NeuroSense, which is a fiber bundle-based kind of miniature microscope where really all the optics and imaging side are off the head of the animal, and you're really imaging through this coherent fiber bundle. It allows you to do multi-region imaging. So that would be the other kind of big commercial options, along with Inscopics and Doric. And then like really everything else listed here is in some one way or another open development and developed in academia. And I think almost the entire bottom row uses some component of the UCLA Miniscope platform. And it's just really great to see all this active development in all these different areas. So hopefully it gives some motivation for if you're our grad student or a postdoc or just starting up your own lab. Incorporating some of these technologies now should just be at the start of what these types of things can do in the years to come. And so we are absolutely not the only people doing development here. And there's, as you saw in the earlier talks, wonderful groups doing amazing work in the field. Cool. So with that said, we're going to go through just some of the newer developments that we're focused on. So we've talked a lot throughout this workshop about the currently released miniature microscope that we developed, which we call the version 4 Miniscope. And a lot of the developments we are working on now are built around this platform. So a few years ago, when we had the version 3 Miniscope, we saw some kind of fundamental limitations of how miniature microscopes were being built. And so the version 4 allowed us to sort of rethink from the ground up the design of these miniature microscopes. So things like moving away from GRIN lenses when you don't have to, so no longer using an objective GRIN lens, moving away from mechanical movement. The moment you put this on an animal, you really want to avoid kind of interacting with the animal as much as possible. So something like incorporating a electrowetting lens seems like a great idea. This was really inspired years ago by Emily Gibson's group that was doing, I think, building their own for a head-mounted 2-photon microscope. Always making things smaller and lighter and really expanding the field of view when possible is also important. So taking this as kind of the building block, we've added some other features. So I think I didn't show this yesterday, but we have this tunable lens. I guess I showed in the kind of real-time software acquisition portion, but you can, a single Miniscope, you figure out what focal plane works on each animal. That just ends up being a number. And you just type that number into software. And a single Miniscope now can image across a whole batch of animals by just typing in the specific configuration for that animal. This was half working with my very old Miniscope at my house, but we've included these absolute head orientation sensors on all the Miniscopes we build now. And this just streams out effectively roll, pitch, and yaw of the animal's head or the Miniscope in real time along with imaging. And so this is just holding a Miniscope and that Miniscope logo moves, but that data gets saved also as well. We'll see an example of that soon. I also talked a little bit of one of the motivations for kind of rethinking what the design of a miniature microscope is that a lot of people want to do cortical imaging, and it doesn't make sense to do the old way, which is to always have to implant a large GRIN lens onto the surface of cortex, or with some systems now, you have to implant a relay lens on cortex and then have your microscope sit all the way on top of the relay lens. So we redesigned these optics to have a larger working distance, which allows us to reach into cortex through a standard cranial window prep. And so this is an example of one of these recordings done by Mega Siegel at UCLA and Alcino Silva's lab. This is also, we've swapped out some of the lenses to give us a bit larger field of view. So maybe this field of view is closer to like 1.5 millimeters. And this is just imaging retrosplenial cortex through a cranial window, which is really great. That mouse actually was prepped for a whole batch of 2-photon experiments. And when she was done with those experiments, we threw on a Miniscope base plate and imaged with the Miniscope. But we can also, of course, image deep. So this is imaging medial prefrontal cortex through a 1 millimeter GRIN lens done by Pingping Zhao, who's a postdoc in Hemang Galshani's lab. This is the raw data coming out of the Miniscope. With a 1 millimeter field of view, we basically are imaging the entire surface of that GRIN lens. And then with some kind of minimal processing, we can really see the cells. There's something like 1,000 cells in this recording. And a nice feature of redesigning the Miniscope is that the optics and the image sensor are much more sensitive than previous generations, something like 10 to 15 times more sensitive, which allows us to image longer with less photo bleaching. So the first long recording we did is this 9-hour recording in medial prefrontal cortex. The animal is just in this open field. That's sort of the heat map of the animal. And over these 9 hours, the gray is kind of the behavior of the animal. Animal ends up going to sleep towards the middle end of the recording. But the overall decrease in fluorescence, sort of both the baseline and the size of the calcium dynamics, drop across these 9 hours by something like 8%. And then since then, we've done 24-hour recordings where we get even less photo bleaching, like 4% across 24 hours. And the quality of recording at the beginning here compared to the 9 hours later is really comparable. And so these newer miniature microscopes with their kind of more optimized optics and more sensitive image sensor are sort of getting us to the point where we can start thinking about uninterrupted continuous recording, right? And I think this has been potentially possible before now, but if you had a system that you had to head fix an animal and do this under a tabletop microscope, it's not feasible to head fix an animal for 3 days uninterrupted, right? So you really have to move to freely behaving animals. And as we can kind of push the sensitivity of these systems forward, and Caleb showed this in his talks as well with the back illuminated image sensors they're using, we're really right at the edge of no longer needing to worry about photo bleaching in many preps. Okay, so we also mentioned before, we've done a lot of work in minimizing the tethering between the animal and the acquisition hardware. This is really important to get natural behaviors Open EFIS with their EFIS head stages also have shown really how critically important this is for a lot of behaviors that they're interested in. But there are certain environments or behavioral tasks where having a tether at all gets in the way. And so we've developed these wire-free miniscopes. The first version of this, again, we call it the version 3 wire-free miniscope, has been available for a few years. And this recording, I think, was done when Tristan and Denise and I were all postdocs at UCLA, and I think Tristan ran this experiment. And this is a mouse wearing one of our first versions of the setup, running on a 25-foot or like 7.5 meter long linear track. The track is so large that we have to put a behavioral camera on each end of the track to see the animal's behavior. And we can record for about 25 minutes with equivalent imaging quality as a wired system. And then here we process the data and pseudo-colored the neural activity here. And there's something like 561 neurons detected. And this is the task that you just couldn't do really if the mouse was tethered, right? It's hard to conceive of a cable management system in which the animal would naturally run on 7.5 meter long track. Here we are looking at hippocampal CA1 neurons. And so we can look at this data a little bit differently. At the top, this is also the same behavioral video, but we've removed the perspective distortion. And so that's the mouse running back and forth on this long track. And then we just pseudo-color coded the cells that are significantly spatially modulated. So things we call place cells. Out of those 550 cells, about half of them are significantly spatially modulated, which are shown here. And we've color coded them just where their peak neural activity rates are. So as you can see, the mouse runs from left to right. The neural population sort of changes colors from blue to teal. And you can see the sequence being plotted in the middle. And then when they run back, you see this other sequence of activity of cells that are yellow thread. And what's cool is you just really couldn't do this with a tethered system. You might also want to do experiments that are in 3 dimensions that kind of tether cable management is hard. Or maybe you want to do social experiments with multiple animals wearing miniscopes. In that case, you really can't do that with tethered systems because the cables get twisted together. So wire-free miniscopes can be useful in a lot of these cases. And as I said, that previous generation system has been shared for a while. I think including the battery, it's like close to 5 grams, which is a lot for a mouse, but a mouse can handle that weight. And that gives you around 25 minutes of recording. And we're just finalizing this newer version, which is built around the version 4 Miniscope, which is a bit lighter, should be more power efficient. And the design is pretty user-friendly to be able to do these wire-free recordings now with the version 4 Miniscope. So this gives us a larger field of view. Potentially for the same weight, we can record longer. It has an option to be optionally wired. So right with the previous generation systems, the general workflow would be that you would attach a wired miniscope to your animal, figure out how bright the LED needs to be, if you need to increase the gain of the image sensor, and then you effectively transfer those parameters to the wire-free version. Because it's not wireless, we're not in real time streaming a video stream off the animal. So you kind of are going into it blind. Once you kind of get the workflow working for you and you figure out how to dial these parameters, it's not really a problem. But it would be really nice for the same miniature microscope that's a wire-free microscope to really just plug in a small cable and turn it into a wired microscope to be able to see the cells in real time and set the parameters you want, unplug it, and let the animal run wire-free. So this new version can do this. While it's wired and plugged in, it has an onboard battery charger, so it will charge its side-mounted LiPo battery. It also has an infrared receiver, what you would find in like a TV or a DVD player, where a remote control that could be just a handheld remote control or something controlled through the computer can send digital bytes over infrared to the miniscope. So things like triggering it to turn on or off are now possible fully wirelessly. This ideally will be done by the end of the year and should be shared soon after. It's like a direct swap out of a version 4 miniscope. So the entire body is the same, the optics are the same, you just change the image, the electronic circuit boards, which is really nice. We've also, for the last few years, worked on an electrophysiology integrated miniature microscope. So the first version of this is this version 3 Miniscope with this like daughter board that incorporates a 32-channel Intan EFIS chip. And then that green board has a very high density connector that can go and connect to whatever types of electrodes are implanted. So we did a lot of the initial piloting of the system, reporting from silicon probes designed and produced by Satiris Masmanidis' group at UCLA, which I'll show in a second. We've also done some recordings from a group of tetrodes in rats. So the hardware is somewhat agnostic to the actual implanted electrodes. And as long as there's an adapter to go from the connector on the EFIS board to the kind of electrode interface board, you should be able to connect to any electrodes being implanted. So some of the first recordings we did, this is now a couple of years ago, this is a mouse wearing this electrophysiology miniscope, or we sometimes call it the e-scope. This just seeing the bright LED on the animal's head running back and forth on a linear track. And then this is not the best miniscope recording, but there's a calcium dynamics in the top right. And then there's a couple channels of EFIS recorded from this 32-channel silicon probe. And you can see these nice downward going extracellular spikes as the animal behaves. In rats, we have a lot more space to work with. In Pat Blair's lab, this was, which their experience in the past had been really doing a lot of tetrode recordings in, let's say, hippocampus, and now they've done some amazing imaging as well. But when they first started getting up and running with imaging, they made a drive they call an oval drive, which is open source and you can find online. And it can mount a miniscope. In this case, we're imaging CA1 in one hemisphere, and they have 8 tetrodes implanted in the other hemisphere. And this was sort of like where imaging in rats was a few years ago, where there's maybe 50 cells here. And I'll show what we can do now in rats. And then in the EFIS, there are potentially a couple extracellular spikes, but what we're really seeing here is LFP as the animal moves. So as you watch the rat move, you can see nice theta oscillations and gamma oscillations show up on these channels, which can provide a lot of additional nice information to go along with calcium dynamics, right? So calcium doesn't give us access to LFP and EEG oscillations. So even having a couple channels of EFIS can be useful. The big challenge here is not really the electronics in designing a system like this, but it's how do you fit everything on the head of a mouse? So the moment you go to something bigger like a rat, there's some advantage of kind of integrating all of this into a single setup, but really you have plenty of space, and rats are large and strong. They could handle a completely separate EFIS system and cabling to a miniscope. So our goal really here is to build something that's very usable for mice. I maybe didn't mention, but also when everything we sort of integrate into the miniscopes, we really aim to not add any additional tethering. So this EFIS system is powered and communicates through the same single coax cable that's already handling the miniscope imaging data. It just packs the EFIS data alongside it. So the setup we're finalizing and hopefully works well enough for us to finally share is a version of this that can be retrofitted to any previously made version 4 miniscope and adds 32 channels of EFIS that can be sampled at 20 kilosamples per second per channel. And the big challenges are twofold. So one is just space, like how do you fit a miniscope base plate and some electrode implant on the surface of the mouse that is reasonable. But that can be solved for many configurations of brain regions. And then another big challenge is that the miniscope gets removed from the animal's head when you're done with your recording for that day. But there's some components of the EFIS setup that needs to stay chronically implanted on the animal. And so when you come back to record every day, you have to make an electrical connection between the miniscope that gets temporarily attached and the chronically implanted EFIS devices. And this has been really challenging when you have an awake behaving mouse trying to connect high density connectors without accidentally pulling the electrodes and damaging them. So our solution, it's hard to see here, but effectively will be a modified base plate that already has the electrode connection built into the base plate. And when the miniscope slides into the base plate, there are spring-loaded connections that connect the EFIS interface to the miniscope. So while it still can be difficult to kind of figure out how you want to fit everything onto the skull of a mouse, likely, hopefully once everything is cemented in place, all that's involved is the standard thing you do to put a Miniscope in a base plate, and it will on its own automatically make electrical connections. So we're finishing this up. Again, hopefully it will be openly shared in the next few months, 6 months or so. There's been a lot of questions, especially in our breakout rooms, about a dual color or 2-channel miniature microscope. And so both, definitely in a lot of collaboration by both my group and Denise Cai's group with, led this project by Phil, which you just heard from, have kind of simultaneously built 2 versions of a dual color miniature microscope. So the one that we've been focused on is pretty simple modification of a standard V4 Miniscope. So this is almost all the same optics as a standard V4 Miniscope, almost all the same housing components, but the filter sets have been swapped out to allow for dual excitation light to pass through the filters. And the circuit board is slightly different, which allows us to have 2 closely packed LEDs on the same circuit board for dual excitation. This was, so I think the hardware of our system works exactly how we expected. And we're now kind of working through the validation steps to figure out what combinations of fluorophores not just can be imaged by the system, but that are like useful in actually getting to be expressed in vivo, which ends up being a bigger challenge than expected. But there's some very preliminary data imaging GCAMP simultaneously with, I should have written it here, I think M-Cherry, but it could be TD Tomato in CA1. This should be available, I think, somewhat soon, but we really are still working through validating it. Maybe I already said this. But it really is a direct swap out of a V4 Miniscope. So if you're interested in ending up doing 2-color imaging, what our suggestion is now is to like not wait for these to be open source, but to use a green Miniscope, a standard Miniscope, and you can get a second Miniscope and just purchase different filters to convert it to a red Miniscope, and then really work on dialing in the viral expression in your animals and image with just a green Miniscope, and then keep that out and image with your red one. And then that, I think, is a bigger challenge than what some groups expect it to be, especially with single photon imaging. And then once that's working, you can just throw in the dual color Miniscope and it should work. I should mention that the way this system works is it does something called color sequential imaging. So there's only one image sensor, and on any one frame being exposed by the image sensor, we have the ability to control which excitation light source is turned on. So when we turn on our blue excitation light source, those frames are going to collect green fluorescence. And when we turn on our greenish excitation light source, those are going to record red fluorescence. And there can be, depending on the fluorophore, a tiny bit of crosstalk between the two, but it's not much of an issue. This also then gives you imaging and optogenetics for free, because you now have one wavelength to image and the other LED to do optogenetic stimulation. The challenges there is that most opsins and calcium indicators have a decent amount of crosstalk in their excitation spectra. So the standard approach you might want to do, and this is what most people that have an end view system from Inscopics do, is they image GCAMP, and then there's these very limited preps you can do to get some kind of opsins into the field of view that will minimize crosstalk between these red shifted opsins and GCAMP. What we've started working on, and we started a long time ago, is to, instead of use imaging GCAMP, to image RCAMP, and then use standard blue excited opsins, and those excitation spectra should overlap a lot less. So that's one of the versions. And then, as I mentioned, in Denise Cai's lab with Phil, they've been working on another type of version of a 2-channel miniature microscope, initially really developed to image some fret sensors. So for fret imaging, you want to excite this fluorophore and then image 2 separate wavelengths, and the ratiometric change, the relative fluorescent change from one of the wavelengths to the other, tells you something about the quantity that you're measuring. And so initially this was built in sort of a large setup from a version 3 Miniscope. You can see kind of the cross section is really like just a big version 3 with a second excitation setup attached to the side. And they got some good fret recordings. This was, and then since then, since then they've also made a version 4 version of this. And so a big difference between what I showed of the previous true color miniature microscope and this version is the previous one has to do sequential imaging. We can never image and be able to separate two fluorophores simultaneously. We really depend on which fluorophore is being activated or excited based on which LED is on. But in this case, they use a different approach. They use a single LED that can excite 2 wavelengths, and then they split the optical path, the emission path, so that one wavelength goes to one image sensor and the other wavelength goes to the other image sensor. So this is effectively a version 4 Miniscope that's a bit taller and has a second image sensor and filter on its side. And now simultaneously, they can separate 2 wavelengths and then align those. So this is an example of just imaging a red and green test slide. I mean, it's the same test slide under the 2 different image sensors, and then the overlay of the 2. And you get, without too much work, really nice alignment between the 2 image sensors. And then here's an example doing the simultaneous imaging of GCAMP and TD Tomato. And you can see green cells, red cells, and then the overlay, those yellow cells are cells that are expressing both. I think in both these cases, the challenge is not so much anymore the hardware. It's really figuring out how to get the expression working well for the combination of fluorophores you want to image. Like one major thing to consider if you think about doing this is that GCAMP, or any calcium indicators, we benefit from it not just being kind of being able to see it spatially, but we benefit a lot from the signal changing in time. This helps us be able to extract what a cell is from background fluorescence and help us identify 2 cells that are different next to each other. When you go to like a structural reporter that's labeling a certain cell population, those are stable in time. And so dealing with the cells that are in focus compared to the background fluorescence that you might get from cells that are also labeled with TD Tomato outside of the focal plane becomes more difficult because we don't have this time varying signal to help identify where the cells are. This is not an issue with 2 photon. 2 photon, you just optically section where you're interested in looking, and you don't worry about out of plane fluorescence. Doing this for single photon imaging in vivo can be challenging because the background is hard to deal with if it's bright relative to the cells. But we're all working on this. I think it should be available soon, and really I think just ends up requiring a final bit more validation. But other things we're working on. So something we haven't really talked about at all, because this talk has been really focused a lot, in general, on just miniature microscopes and oftentimes in freely behaving mice. But over the last few years, we've been developing a larger field of view miniature microscope. This work was done by a postdoc in my group, Cheng-Lang Guo. And the goal here is the moment you move away from mice, you get a lot of space and weight to work with. So, mouse, let's say, comfortably can hold 3 grams, and they really can hold up to 6, but people like to keep it under 3, let's say. A rat is like comfortable up to 25 or 30 grams. And some people throw on a lot more weight than that. So if we have 10 times more weight to work with, we can theoretically design more capable miniature microscopes once you move away from these small animals like mice. So this is where this idea came in. And from the first slide that I stole from Piho and a bunch of other people's work, other people are working on this. Oftentimes the push is to go to large field of view without single cell resolution. So this kind of like mesoscale imaging with non-single cell resolution. Because of a lot of the research we're interested in, we really are always mainly focused on building tools that can resolve single cells. And so this large field of view Miniscope, which we just put up on bioRxiv, can achieve actually a field of view of like 3.5 by 2.5 millimeters with single cell resolution across the entire field of view and weighs under 14 grams. it follows all the design principles that all the other miniature microscopes we built have followed. So it's open source and designed actually to be usable, so and understandable. So we're working on having open source distribution companies starting to source and put together assembly kits for these systems. But a takeaway is like this system actually works and works reliably and really well. And I'll show some data from the system soon. But it has an objective module, very similar to what we saw with the version 4 and the assembly of the version 4. This objective is just a larger and can incorporate a couple more optical elements. And then the main body is also similar to the version 4, but we have more space to correct for some of the optical aberrations. It has a tunable lens inside. And then the image sensor and LED are flipped. So the image sensor sits on the side in the back and the LED sits on the top to lower the center of mass of the animal. This slide shouldn't be here. Okay, so give you a comparison of what you get from the field of view. So this green rectangle is sort of the average field of view you get from our old version 3 Miniscopes. This orange circle is the field of view that you generally expect from the standard version 4 Miniscope. And this large field of view Miniscope does quite a bit better. So this is imaging in a head fixed mouse through a cranial window. And you get a much larger field of view. And again, we resolve single cells across the entire field of view. That recording is done in this head fixed mouse, which is running on a circular treadmill. And just an interesting piece of kind of interesting fact here is we were just trying to do some really simple analysis to see if we can pull out individual cells that were increase their activity during bouts of movement. And those are these red cells here. They significantly increase their activity when the mouse moves. And it just turns out that there's also this subset of cells in green that has increased activity when the animal sits at rest. But if you do the same analysis without single cell resolution, so if you sort of neither imagine deep downsampling your spatial resolution or kind of blurring it, you lose the detection of all these cells that get suppressed when moving or are more active during running. And that's because the rest of the population and the out of plane fluorescence kind of overwhelms them. But if you can get down to single cell resolution, you can start to see these smaller populations of cells activity that sort of get lost when you can't do that. But we also want to do freely behaving experiments. So in collaboration with Tad Blair's lab, who again, I showed that eScope recordings they did a few years ago, and now moved on to using these large field of view miniscopes in freely behaving rats. We get to image, in this case, an entire surface of 2 millimeter diameter GRIN lens. So where before they were getting like tens to maybe 100 neurons imaged in CA1, now they get the best animal we have has 1,300 active neurons detected through this GRIN lens in a freely behaving rat, which is really wonderful. This comes from having a larger field of view. It also comes from the system being substantially more sensitive than the version 3 Miniscope and even more sensitive than the V4 Miniscope by about a factor 2. The experiment, one of the experiments we ran is this rat that's running on this large rectangular track. We can track the position of the animal with just a behavioral camera. And because we have also this head orientation sensor, we know in real time or in this recording, the actual head orientation of the animal in roll, pitch, and yaw as the animal's running. And for this session, there's something like 1,300 active cells recorded in CA1 in rats. And around half of those are significantly modulated based on the spatial location of the animal or place cells. And so we're plotting those 600 cells activity in the middle. And you can see these little nice sequences of activity when the animal runs down these different arms. And what's really great, it's not just about like, okay, we get more cells, but for like Tad's lab and Garrett Blair, the grad student who's now a postdoc that did these recordings, it's not just about getting more data, but it's really fundamentally changed the type of questions they can ask. When you can go from having tens of cells or maybe 100 cells to having 1,000 cells or more, it opens the kind of types of scientific questions and analysis you can do. They can now start performing much more interesting like decoding analysis on the neural populations. And especially if you want to look across recording sessions and look at cells that are active across multiple sessions, as you do that, especially in hippocampus, you start cutting out the population you record quite a bit if you're looking for cells that were co-active across 3 sessions. So the more cells you can record, kind of enables the more type of science you can do, which has been great. We'll just spit that some of the data from that session. And then kind of a side thing at the time was that we thought, well, rats can still handle more than 14 grams. So can we make a new type of acquisition board that just can convert any wired miniature microscope into a wire-free one? This board is too heavy for a mouse, but sort of anything bigger should be fine. And so this board that mounts to the side of this large field of view miniscope effectively replaces the wired DAQ that connects over USB to a computer, and does everything on this board. It's battery powered, logs data onto an SD card, but just connects over a coax cable. So the same board can connect to a V3 Miniscope or a V4 Miniscope, and then just turn the system into a wire-free setup. You're working with rats. Rats are happy to carry even like a backpack with a battery in it. And SD cards now have a ridiculous amount of storage capacity. So you're really no longer even limited to like a length of recording because you're going to run out of storage or power. And you really can record for as long as you want, as long as you're not photo bleaching. So also this, we also just, both the large field of view Miniscope and this wire-free expansion board, just recently, we put all the documentation and stuff on GitHub and the repository and wiki. In this wire-free version, you get the same imaging qualities, right? So the same wired Miniscope, but connected to a different acquisition board. I don't have a video for this, but in this case, we are just looking again at CA1 pyramidal cells as the animal is moving around a small open environment. And these are just a few examples of place cells firing along the animal's path for 20 minutes or something in this open field. The top is the behavioral paths with colored dots representing locations where these cells were active. Below those, effectively their place fields, and the red outline sort of outlines the edge of those place fields. The same postdoc, Cheng Lang, when building this large field of view Miniscope, had the idea of like, why don't we try to make one that also fits on the head of a mouse? And he did exactly that. And it surprisingly works extremely well. So this is effectively a version 4 Miniscope that is slightly elongated. It uses the image sensor from this large field of view Miniscope. So this higher sensitivity, higher resolution, larger field image sensor, but can be put into a package with slightly different optics that is light enough for a mouse. So this is 3.3 grams Miniscope. It's still smaller and lighter than the old generation miniature microscopes, and can resolve single cells across its entire field of view, which is something like 3 millimeters in diameter. So this was the absolute first recording we ever did in this cranial window, again, looking bilaterally at retrosplenial cortex across the midline with help of MEGA. And I don't know how well it shows up over Zoom, but there's a bunch of tiny little dots flashing, which are individual cells. And then in collaboration with Ping Gulshani's lab and Ping in his lab, she does a lot of imaging of medial prefrontal cortex, and the field of view is large enough to look bilaterally across 2 implanted GRIN lenses. And Ping is somewhat of a wizard with surgeries and GRIN lens implants. And so she's managed to, across actually quite a few animals now, implant 1 millimeter diameter GRIN lenses bilaterally over medial prefrontal cortex and get their focal planes above the lens pretty much coplanar, so that at this single small large field of view Miniscope can image both GRIN lenses simultaneously. So the actual recordings are this entire field of view, but we can kind of crop out and process just the area around these GRIN lenses, and we get, again, single cell resolution bilaterally with a single 3.3 gram miniature microscope, which is pretty cool. I think I have just a couple, maybe one more project to show. So this is something that we've been kind of working on over the last few years, but we are really kind of pushing forward with it now, is to take the design of a miniature microscope, which is generally really focused on epifluorescent imaging or wide field imaging, right? We have a LED or light source that effectively gets defocused through the optics of the miniature microscope to uniformly or near uniformly illuminate the tissue under the microscope, and then we just collect fluorescence from everything that gets excited under the microscope. But when we, a few years ago, were thinking about redesigning the miniature microscope for the version 4, we already had ideas of maybe wanting to be able to like structurally manipulate the excitation light. And a key feature or way to do that is that the excitation light of the V4 Miniscope now goes through all the same optics that the imaging does. So you can see this blue path here. You forget about the half-ball lens up on the top, but it goes through the achromatic tube lens, the tunable lens, and the lenses that are part of the objective. In the standard configuration for the V4 Miniscope, this just means that we have a lot of space to defocus it really well and give this uniform illumination. But we can do a very simple modification without changing much of the optics. And instead of just using a single excitation light source, we can take that excitation light source and reflect it off a spatial light modulator. So either a DMD, which is like a chip with a bunch of tiny mirrors that can flip on or off, or a liquid crystal on silicon display. But you can think of these effectively as little chips that are able to reflect a pattern of light. And these are used in projectors or like augmented reality goggles. So now we can illuminate the surface of a spatial light modulator. And based on the pattern that's driving the spatial light modulator, we can generate a pattern that gets focused coplanar in the brain, coplanar with where we're imaging, right? So now we can project an image excitation pattern in the brain coplanar to the plane we're imaging. So this is one of our first tests in just fluorescence brain slice. And we are projecting through our onboard projector cross pattern and imaging that, right? So the actual population of neurons is shown in the top right. And now we can, at actually hundreds of frames per second, modulate the spatial excitation pattern in the brain. We can do, when we're not worried about scattering, we can project pretty high resolution images. This is the UCLA SEAL. We can also do color sequential excitation. So on even and odd frames of the projection system, we can project different wavelengths. In this case, we're just projecting different components of the UCLA bear. We can also image cells and then be able to generate an excitation mask that chooses to excite or shine light on a subset of cells. Again, without imaging too deep, we should be able to achieve single cell resolution. As you go deeper, the excitation light scatters, and then you get kind of near single cell excitation abilities. I'll skip over this, but one advantage is that we can do really interesting things with the excitation path to kind of address what a lot of the fundamental limitations there are with standard miniature microscope imaging. So we can offline do some somewhat optical sectioning or remove background fluorescence. And we can do other things. But a really quick example is this is just a head fixed mouse imaging through, I think, cortex, cranial window, and a comparison of in the same mouse imaging just a wide field illumination versus just a very quick generation of like an excitation mask where we focus light on a subset of cells, and we get a significant increase in signal and signal to noise. But you could imagine the more exciting thing here is to be able to improve the imaging side of things, but while doing that, be able to use a second excitation light source and be able to optogenetically control cells across the entire population you're imaging at hundreds of frames per second at a single cell resolution ability. If you can do this and then couple it in with real-time closed loop processing, you can start developing systems that can be based on models that make a prediction based on a model and make a prediction of what would happen to the behavior or to the neural dynamics when you perturb the system. And then you do those perturbations either through behavior or through optogenetics, and you can test the model in a closed loop fashion across the same population of cells in real time, which is exciting. The final thing is like, we don't just build miniature microscopes. And I think this entire field that is focused on kind of pushing what can be done in freely behaving animals, everyone understands that it's not just about the wearable neural recording device, but it's about the entire ecosystem that you need to actually do these experiments. So a couple months ago, we released this thing we call a mini cam, which is a pretty simple behavioral camera, but is kind of equivalent to what you buy from FLIR or these other, like $500 to $700 USB cameras, but a bit cheaper. These are sold from OpenEPA's production site, but there's supply chain issues, as with everything else, so I'm not sure if they're available right now. And then, as I showed yesterday, we're always working on improving our software. So we try to make it really kind of do what you need to do for experiments. But if people have suggestions or features that they would like to add or issues, please, let us know in the Google groups or type up an issue on GitHub. And then finally, as we saw from Phil, it's not just about the hardware and acquisition, but it's really about the processing side of things. It's not just us working on it, it's a bunch of other people working on it. And I think there's this kind of view as we move to the future to really start enabling this entire workflow all in an open source way that would really accelerate the development of these tools and the science that can be done with it. Oh, almost right on time. So I'll end here. Thanks to everyone in my lab, Denise and Tristan, from the initial developments that we all did for miniature microscopes up to developing new tools now and testing them. And Phil, of course, that's done Minion and the 2-color Miniscope. And then all the collaborators also at UCLA, and especially Pimon, Alcino, and Ball, who continue to actively be part of the Miniscope project, but also gave the initial vision of not just building a tool for themselves, but to try to kind of really invest even before there's funding for it to build a tool that can benefit everyone. So I think with that, that's my last slide. We are pretty much at the end. I couldn't see any questions. If there are any, I guess we have a couple minutes before we do kind of the closing remarks with Tristan and Stephen. Oh, there are a few questions. That is a lot.
There were a bunch of questions in the chat for you, Daniel. I think for time considerations, maybe we'll take one right now, and then you can go through the chat and answer them in the next few minutes. And then we just need to wrap up in the next few minutes. So let's take one question from Nate.
Yeah, go ahead, Nate.
Yeah, I guess I'm wondering if the STIMscope is conceptually similar to a pinhole confocal in the end. Is that sort of what ends up? And then my second question is just clarification. The electrophysiology board you said can basically be agnostic for any input. So it could be an MUA fiber, it could be the silicon probe, it could be EEG. Is that correct?
Yeah, so basically anything that could be sampled with an Intan eFIS chip can be sampled. I mean, it's effectively just built around an eFIS chip. So anything measuring electrical signals in the brain that are on the range that those eFIS chips function, usually plus minus 5 millivolts at, I can't remember the bit depth off the top of my head, 16-bit depth would work with the system, and you just need an interface connection. And we've made a bunch for different tools. And for your first question, yeah, it's definitely not the same as a pinhole confocal, but using a different approach, we can achieve potentially similar things. And so one thing we've piloted is the pinhole confocal ends up effectively blocking out fluorescent lights that doesn't come from your focal plane. And you can achieve something similar by a couple different approaches. And one is this structured illumination approach where you're scanning these high frequency bars across the focal plane of your field of view. And then with a really simple computational offline algorithm, you can effectively kind of throw away out of plane fluorescence. They sort of get canceled out. So you can start to approach the ability to potentially remove some out of plane fluorescence and maybe even be able to image multiple planes with different cells with some of these other approaches. Yeah. Cool. Any other questions? Again, if there's something interesting, you can email us or post on the Miniscope Google group as well, which would be great.